So like she said, I'm Tracy Colin. I'm a PA in GYN oncology. I'm going to talk about common and uncommon mutations associated with our gynecologic cancers. Mostly we're going to be talking ovarian and endometrial in these, but there's a few other things that can get involved. Objective-wise, we're going to identify risk factors that are associated with increased incidence of a GYN diagnosis. Characterize what these germline mutations are, and discuss, who to test with these mutations. So I'm going to talk about BRCA1 and 2, or as a heavy hitter everyone thinks of, Lynch syndrome, and kind of how that correlates with microsatellite instability, which is kind of a big buzzword right now in oncology with immunotherapy. Then we're going to talk about some less common mutations such as BRIP1 and RAD51. Kind of go through, if you get a testing result, you may get things like a variant of undetermined significance or no mutation found, but the patient has a family history, and what do you do with that kind of information? So I include this slide only to prove a point that it's very complicated. So you got your BRCA, BRIP1 comes in here even though it's not on the chart, your RAD51, Lynch is over here, T53. So everything's kind of all related with all these mutations, and it's all interacting with each other, so it can get pretty complicated on what causes what and how these are interacting. But this whole pathway is called the FA-BRCA pathway, so you might hear that sometimes when people are talking, especially about like BRCA and BRIP1 and some of the breast cancer genes that aren't associated with ours, but they tend to come in that pathway a lot. So general facts to give some baseline risk on these cancers as we go forward, so you can kind of compare. So in ovarian cancer, there's about 21,000 cases a year and 14,000 deaths, so pretty close numbers there. Your general population risk is about 1.3%, and we'll come back to that number a lot as I talk about risk with the other mutations. And average age of diagnosis, 63%. Overall, with all stages surviving 5 years, is 45%. Ovarian cancer tends to be diagnosed later stages. A hereditary cause is found up to 30% of ovarian cancer, depending on what study you look at, so a pretty significant amount. Your high-grade serous and endometrioid cell types are going to have the higher burden. Low-grade serous has a little bit. Mucinous, they think, might not really even have a genetic risk associated with it. And also for the purposes of this talk, when I'm saying ovarian cancer, I mean ovarian, fallopian tube, or primary peritoneal, unless I state otherwise. They're really the manifestation of this different manifestations of the same disease. In endometrial cancer, there's significantly more cases per year, 63,000, 11,000 deaths, so those numbers are much further apart than in ovarian. Almost over twice the risk as a population than ovarian, 2.8%. Average age similar at 60%. Surviving 5 years, if you look at all stages, is 81%, because many of these are caught in much earlier stage. However, if they are advanced stage, it's only 16%. Hereditary cause is found in about 2 to 5%, so not as much as ovarian, but still pretty significant. And with the mutations in endometrial cancer, BRCA and Lynch account for the majority of these hereditary causes, whereas ovarian, it's a wider spectrum. So first we're going to talk about BRCA1 and 2. So almost all these genes are obviously going to be tumor suppressor genes. So normally BRCA fixes mutated DNA using homologous recombination repair. So sometimes people refer to things with this mutation by its repair. But if it's abnormal, cell mutations add up, cells proliferate incorrectly, increasing your risk for cancer. With BRCA1 and 2, we know a lot about these genes compared to a lot of the others. So they're very long genes. You can have mutations at various nucleotides within the genes. We know that there's at least 6,500 mutations known to happen within these genes. And sorting through which of those cause an actual phenotypic high risk and which don't isn't determined for all those genes yet. So that can complicate test results as well. But we know more and more all the time. BRCA is an autosomal dominant inheritance. It's highly penetrant. And your lifetime risk with BRCA1 is 40 to 59%, so pretty high for ovarian. BRCA2 is lower at 35%. So these are kind of classic hereditary pedigrees for the two. So with BRCA1, you see that you're looking just at ovarian and breast, and you're looking at females. With BRCA2, you have males involved, you've got a prostate cancer risk, you've got a pancreatic cancer risk. So although we see less percent BRCA2 developing ovarian cancer, they've got more kind of to worry about. There is questions on whether BRCA2 might increase your risk of other GI cancers and melanoma. So these are the NCCN guidelines for who to screen for BRCA1 and 2. So the top is kind of going through people with more of a breast cancer history or breast cancer family history. So that's pretty detailed and specific on who to test and who not. Much easier in our field. If you have ovarian cancer, you should be tested. So pretty straightforward. And family history is actually pretty straightforward. If you have a first or second degree, just one person with ovarian cancer history, you should be, and they can't be tested or don't want to be tested, you meet criteria to be tested. So that's a lot of people and pretty straightforward. It also gets in kind of with the pancreatic and prostate if you don't have a lot of females for the BRCA2. And it's also estimated, so about only 20% of people who qualify by these guidelines get tested. So it's a huge patient population we're not getting tested, and that you could probably prevent 655 cases of ovarian cancer per million population if you actually followed the guidelines. So the two mutations have differing age of average diagnosis. So BRCA1, there's pretty minimal risk before 35, but from 35 to 40, about 2 to 3% of people will develop ovarian cancer with this mutation. The overall average age is 50. BRCA2, your 2 to 3% will develop by age 50. But the average age is 60, so kind of 10 years older for 2 versus 1 in all accounts. Interestingly, if you do have ovarian cancer, your progression-free survival and overall survival is a bit higher if you're BRCA2 followed by BRCA1, followed by having no mutation. So we can modify risk for ovarian cancer and BRCA1 and 2. And this would be by performing a bilateral salpingo oophorectomy or taking out both tubes and ovaries. I won't get into it too much, but it's thought that these diseases probably start kind of at the very ends of the fallopian tube, regardless of whether we call it ovarian or fallopian tube cancer. So that's why you want to take that tube, is almost the more important part. And there's growing evidence that the risk-reducing surgery, by taking out that natural hormone, you can mitigate the breast cancer risk as well if they're not having the prophylactic mastectomies. So we can't really mitigate too well the risk of primary peritoneal carcinoma. So that is a cancer that kind of starts in the lining of your abdomen. So there's some studies listed up here about the risk of primary peritoneal after risk-reducing BSO. And it's still about 1.7% total risk. So kind of getting close back down to your baseline population risk for ovarian, fallopian tube, primary peritoneal, the whole thing, but still a risk. So then there's this question of endometrial cancer risk in BRCA cancer patients, still kind of trying to be sorted out. It's mostly thought to just be associated with BRCA1. It tends to be more associated with your high-grade endometrials, which would be like a serous or carcinosarcoma. It's hard to sort out because in a lot of this patient population, they've already had breast cancer, they're on treatment for breast cancer, one of which things is often used is tamoxifen, and tamoxifen does increase your risk for endometrial cancer on its own and for these higher grades. So a lot of the studies, it's hard to sort out the population, get enough people. They've also found there's actually a Jewish founder mutation. So I talked about there's different spots within BRCA you can be mutated. So there's one mutation that they call founder mutation that is not the Ashkenazi Jew mutation that tends to be associated with endometrial cancer. So they're wondering if it's just a couple mutations within BRCA that create this risk. And that's why it's so hard to sort out at this time. So as far as when to do the surgery, so for BRCA1, you'd want to do it between 35 and 40. And for BRCA2, you'd want to do it between 40 and 45. So if you're looking at a BRCA1, and you're taking out their ovaries at 35, that's pretty young to be left without any hormones. So can you give them hormone replacement? The general studies say yes, you can. From an ovarian cancer risk, it doesn't really increase your risk of getting a primary peritoneal or anything like that studies have shown. It's more trying to look at, is that going to increase the breast cancer risk? So obviously, if they have a hysterectomy at the time of their BSO, that helps because now we only have to give them estrogen. We don't have to give them estrogen and progesterone. So that helps mitigate the breast cancer risk. And then if they go through with the prophylactic mastectomies, obviously that also eases our minds on giving them a hormone replacement. So this shows kind of when people with both mutations ended up getting their cancers. So for BRCA1, you can see it's a little more spread out than BRCA2, but still kind of clustered in that 50s and 60s age range is the highest incidence. With BRCA2, it's pretty, in this review, clustered in the 60s. Interesting in this, no one over 70, so kind of, if you make it long enough, you're probably not going to get the phenotypic manifestation of the disease. So, all right, on to Lynch syndrome, which is a little more complicated in some senses, a lot more interactions going on than BRCA. So first I'm going to kind of try to explain microsatellite instability and mismatch repair, because that's kind of the basis for Lynch syndrome. So microsatellite instability, about 20% of endometrial cancers have microsatellite instability. We call that MSI high or MSI positive, but only 5% of those have Lynch syndrome. itself, so the germline mutation. So what exactly is microsatellite instability? And that is a great question for a geneticist, but here's my explanation. So microsatellites are repetitive nucleotides. So you can see AT, AT, AT. Okay? So when something happens in DNA copying that you get an extra nucleotide in there, that is a microsatellite instability. So what happens when you're looking at normal cells versus tumor cells is you have more repeats and variable repeats of these microsatellites compared to the normal tissue. And that's what they're looking at comparing when they're saying this tumor is microsatellite unstable or microsatellite high, is seeing these variabilities in the tumor compared to the normal tissue. We'll talk about the 4 genes of mismatch repair, but the one gene, PMS2, you're going to see kind of doesn't follow the rules as much. So PMS2 has less incidence of microsatellite instability than all the other MMR genes. All right, so mismatch repair. So mismatch repair genes, again, are helping repair problems within the genome that happen when things get copied wrong. So you can see here it can be like a single nucleotide. It can be like a bigger insertion. And generally these 4 genes, MLH1, MSH2, MSH6, and PMS2, form together to fix it. So if you've got a mutation in one of these proteins, you're not going to be able to fix this as well. And then you're going to end up with more mutations in your genome, which is why you have your increased risk of cancer. The most common mutations are the MSH2 and the MLH1. So about 18 to 30% of endometrial cancers will test positive if you test the tumor for silencing of one of these genes, but that doesn't mean they all have Lynch, and we'll kind of get into that. But also 1 to 2% of serous ovarian, 12% of clear cells, and 10 to 19% of endometrioid ovarians. So the endometrioid kind of makes sense why that might go with endometrial cancer, but clear cells always one to keep in mind with Lynch. I did find a study I found interesting that people with these mutations more commonly have higher grade and stage of their cancer, and more commonly have what we call LVSI, or lymphovascular space invasion. They're less commonly overweight, which that makes sense because most endometrial cancers without Lynch are caused by excess estrogen, which happens when you have excess adiposity. So if it's caused by a gene, you wouldn't necessarily be overweight to get it. But overall survival seems to be unaffected, even though they're presenting with more risk factors. But yet, if you look at just Lynch patients and their overall survival compared to all, it's the same. So it's something that's still kind of being sorted out, what might be happening there. So Lynch syndrome overall is autosomal dominant again. And I should say with almost all these mutations, they're heterozygous, so you only have 1 copy of the mutated. Most of them, if you have 2, it's lethal. The phenotype is much less penetrable in Lynch syndrome than compared to like a BRCA mutation, and varies a lot depending on which of the proteins you're mutated in. The incidence is about 1 in 200, so it's a pretty common mutation in our population. And again, the 4 proteins you can be mutated in are MLH1 and MSH2 are your big ones, then also PMS2 and MSH6. So this shows you all your risks of cancers with Lynch syndrome depending on which mutation. So PMS2 has kind of the smallest. Your still lifetime risk of cancer can get pretty high depending on the study, up to 53%. everyone thinks of colon cancer when they think of Lynch syndrome first. So your risk is pretty high across, but not as high for PMS2, and endometrial's risk similar there. If you start looking at your other three, your colon cancer risks can get pretty high, into the high 60%. And your endometrial, with MLH1 especially, you're looking at a risk that's actually slightly higher than even your risk for colon cancer. And still pretty significant for the others, for MSH2, 21%, and MSH6, up to 44%. Ovarian cancer tends to only really occur in the more common mutations, but pretty significant, 20% and 33%. Small risk in MSH1. And then other cancers known to be associated include your other urologic, your other GI cancers of all sorts, basically, some risk of brain tumors, and also sebaceous gland tumors. So Lynch syndrome is 2 to 5% of endometrial cancer cases. So the average age of diagnosis is 45 to 54. They tend to have a higher incidence of being located in the lower uterine segment, so this can cause confusion, especially if you're looking at a slightly younger population. You might get misled down the path of a cervical cancer versus endometrial, so that can be difficult to sort out. The overall, for any of the mutations, lifetime risk of ovarian cancer is 12%. Like the other graph showed you, it's higher for your MLH1 and MSH2. The average age for ovarian is 43. So that's actually, if you think about what BRCA was, younger than BRCA for those ovarian cancer patients. Interesting, under 30, ovarian cancer is not very common at all. And I'm talking serous and epithelial ovarian cancer, not your sex cord and germ cell. So it's not very common to have a genetic mutation, actually, if you get ovarian cancer under 30 overall. But if you do, it's much more likely to be Lynch than it is to be BRCA mutation, which I did not know before doing this. And I'm like, I wouldn't have thought that. So, and about 20% of people with Lynch will get their cancer before the age of 40. So, again, pretty young. So this is kind of a graph showing the different mutations and the age they were diagnosed with their ovarian cancer. So the average age for all of them, even though there weren't any they thought were associated with PMS2, pretty similar, but all kind of early 40s. So who to screen with Lynch syndrome? So these are the SGO recommendations. They're a little easier to read than the NCCN ones, but I'll show you those next. So anyone diagnosed with endometrial or colon cancer before the age of 50 should be tested. And we'll talk a little bit later about screening the actual tumor, which is something we do at this institution. That kind of mitigates some of these, but anyone in that category should be tested for Lynch. And then if you look at family history, anyone with 2 first or second degree relatives with Lynch-associated cancers should be tested. This is the NCCN guidelines. So again, focus on diagnosed before 50, and then they kind of get into tumor testing of MMR and MSI, which I'll explain in a little bit, being a reason to test those patients. So this is a graph kind of showing how screening can work on the actual tumor. So if we know someone has endometrial cancer and we do a surgery or a biopsy on them, you can either test that tumor for microsatellite instability, or you can test it for if these proteins appear to be acting normally. So if you test the microsatellites and it's microsatellite and stable high, then they should undergo testing. At our institution, we do the immunohistochemistry testing for the 4 genes. So if MSH2, MSH6, or PMS2 are abnormal on that screening, they're referred to get tested. MLH1 likes to be a little harder. So if that test's absent, you actually have to do something called methylation. So in patients, especially older patients, this protein can become non-functional by a methylation, even though it's at the somatic level. So if it's hypermethylated, so all these methyls are what's silencing the protein, then it's not Lynch. But if there's not hypermethylation, they should be tested. When you're screening people, MLH1 and PMS2 tend to both be silenced together. You can also have the other 2 coupled, MSH2 and MSH6, but it's not quite as common as the MLH1 and PMS2. IHC screening is about 92% sensitive, so pretty good overall in finding them. But just because you screen them for the proteins doesn't mean they can't still be microsatellite and stable, which can be important in the future if you're looking at immunotherapy. So similar to BRCA, we can do risk-reducing surgery. So study in 2006 looked at women who they just took out their uterus, they took out their uterus, tubes, and ovaries, or they did nothing. Of those who had their uterus removed, no one developed endometrial cancer, versus 33% who did not. In the people who had the BSO with their hysterectomy, no one developed ovarian cancer, versus the 5.5% if we left the tubes and ovaries in for either group. And nothing on anyone getting primary peritoneal after this. So the average age recommended to do this prophylactic surgery in Lynch syndrome is actually at the end of childbearing. So that can be difficult because if someone's 20 and they already had 2 kids and they say they're done, should you take everything out? Well, probably not. But the recommendation is just end of childbearing. There's not a specific age like there is in BRCA at this point. All right, so that's Lynch. So now some of the less common genes, some of the genes we're still learning a lot about. So BRIP1 is a DNA repair gene, interacts very closely with BRCA. It's found in about 1.4% of ovarian cancer patients. BRIP1 is not shown at this point to really have a breast cancer risk. So a lot of people think that every gene that shows up as mutated is breast and ovarian just because BRCA is so ingrained in us. And that's not the case with these rarer ones. There's lots of genes that are just one or the other that we know about now. Lifetime risk for these patients of ovarian cancer is 10 to 15%. Average age of a lot of these less common tends to be close to what we see for just general ovarian cancer. And so the prophylactic surgery is recommended a little bit more like BRCA2 with 45 to 50. Your RAD51C and D mutation, only about 1% of ovarian cancer patients, ovarian cancer cases will have this mutation. You still have a 9% lifetime risk if you have this mutation. So not as high as many of the others, but if you think about the general population being that 1.38%, significantly higher than that. Again, the RAD51, there is not the breast cancer risk. Average age, around that 60, so you'd want to do prophylactic surgery in the 45 to 50 range. Now for the really fun ones and really rare ones. So TP53 mutation is better known as Li-Fraumeni syndrome. So this TP53, if anyone took bio, knows that it interacts with this lovely cell cycle that gave us all nightmares in undergrad. At least it did me. And so when it's mutated, the cell cycle goes crazy. So cells proliferate, and so you have a very high cancer risk. So your risk lifetime for these patients is about 90%. So you're probably going to get cancer, but it's extremely rare of a mutation. So most people think, well, it's obvious if someone has this in their family, right? But you can be the first mutation. You could be the first person to have a germline mutation. That's a new mutation. And there is growing data on mosaics, which are people who have two different sets of DNA within their body, having just one set with mutations, so they don't phenotypically present quite as strongly as the full carriers. So with this, you're thinking actually more sarcomas. So this is one that's a little different. So for us, you're looking a lot at vulvar sarcoma, as well as you can get a uterine sarcoma, and there's some question of some other serous cancers with gynecology being associated. And this is a list of the most common mutations that these patients get. So your sarcomas, brain, breast, leukemia is in there. They're much more sensitive to getting a radiation-associated cancer, which radiation-associated cancers tend to be sarcomas, so that kind of makes sense. So the screen is actually whole body MRI, so you get quite the imaging if you have this. P10 mutation is better known as Cowden syndrome. So similar to BRCA, you can have slightly different spots mutated in P10 and get very different kind of phenotypic presentations. So these patients tend to all have kind of dermatologic and soft tissue, non-cancerous manifestations. So some of the mutations within P10 cause just that. But the specific mutation in P10 that causes the cancer is known as Cowden's. So it is a tumor suppressant gene. It is autosomal dominant. So I have a couple pictures of the dermatologic stuff. So these are little fibromas on the tongue, and these are keratoses kind of on the hand there. So it's about 1 in 200,000 people, so not a very common mutation. Breast is the biggest risk in these patients. Tends to be early. Males are affected with this, but endometrial is about 20% risk. Thyroid's another heavy hitter, along with renal and colon, and the verdict's still out on gastric and melanomas. There might be some increased risk, but it's not as clear. So your risk for endometrial with this starts on average about 25. But there are case reports of people as young as 14 with Cowden's getting endometrial cancer. So you have to keep that in mind. So the screenings are here. So obviously they focus on many different cancers. For us, the endometrial biopsy is kind of starting in your 30s on a regular basis until they're done with childbearing and can have a hysterectomy. Peutz-Jeghers syndrome, so 1 in 8,000, so not as rare as the other two. It's a mutation in the STK11. So when people think of Peutz-Jeghers, they think of people with freckles all over, and kind of the known thing is, the freckles on the mucosa is what it's known for. And then they get these hamartomatous polyps within their GI system, usually by the age of 10 to 30. And that can cause all sorts of gastric and GI cancers. But they also have about an 18% chance of getting a gynecologic cancer. with ovarian being about a 21% chance. They also tend to get a lot of non-cancerous sex cord tumors of the ovaries, which those tend to present with signs of hormonal off, because it can be hormonal secreting. And they actually have a cervical cancer risk, which is a little unique, about 10%, but it's of rarer types, such as adenoma malignum and mucinous adenocarcinoma of the cervix. So not your typical squamous cell, not HPV related, but much more aggressive cervical cancers, unfortunately. So usually by age 40, these patients have 76% of them have had some kind of cancer. So the screening is really just more focused on the GI stuff. So the next few slides, I'm going to kind of go into what to do with some test results that you may get back. Here at Methodist, we have a hereditary people, so they kind of work through all this with our patients. But if you're from outside Methodist and you order it on your own, what do you do with some of these results? So you can get mutations because you're testing anymore a big panel, which has been shown to be cost effective in studies to do the big panel. Is you can get what's called a variance of undetermined significance. So we found a mutation in a gene, but we don't know that that gene causes cancer, but we don't really know what that gene does. So that can be 2 to up to 35%, depending on the study, of getting a variant of undetermined significant on a test result. So the general guideline is that they should be treated on family history alone and not treated that they have any kind of mutation at all. You can also find mutations on the report that don't explain the cancer in their family. So an example would be strong family history of ovarian cancer. You do a panel, they have a mutation in ATM. ATM is known more for pancreatic and breast cancer. There's no pancreatic or breast cancer known in their family. Well, what do you do with that? So are they the first to get that mutation? Should you test someone the generation back to know if it's hereditary? If there's been no phenotypic presentation of it, do you need to do anything about it? So counseling before you test the patient that these things can pop up and we might not know what to do with them is important. And then I've kind of listed other mutations that kind of come up that are not at this point known to cause a significant increase in gynecologic malignancies. A lot of these cause breast cancer risk, but not anything in gynecology. So the next thing you can get on a test result is someone could have a very strong family history, multiple members with ovarian cancer. You do the panel, and there's nothing. So what do you do with that kind of result? We call that an uninformative result. Some possible reasons is you could have a mutation on BRCA1 or 2 that hasn't been identified as of yet. It could be a gene that's so rare we haven't identified it. Those patients in the family could have had sporadic mutations versus germline.
White Health is giving flu shots in the Centennial Room till 3 p.m. White Health is giving flu shots in the Centennial Room till 3 p.m. Thank you.
And maybe some of these variants of undetermined significance are found later to be significant. So if you have one first-degree relative with a history of ovarian cancer, but everyone has negative genetic testing, your lifetime risk is elevated from that baseline of 1.38. It's about 5%. So you can consider doing something about that. Oral contraceptives can mitigate your risk. And you can consider risk-reducing BSO, but that can be difficult with insurance companies. So this is just an example of a test result with BRCA. So it's telling you they have a mutation BRCA2, then it gives you all this mumble jumble. So basically all this is telling you is that this is the actual nucleotide number that's mutated. It's the 8,435th nucleotide in the gene, and that make sure I get this right, a guanine has been switched to thymine. And this is the protein it's supposed to code. And it'll always say heterozygous, because like I said, if you had 2 copies, that would be lethal. So that's just meaning it's only 1 copy in your genome. So like I kind of said before, there's at least 6,500 different spots in BRCA genes that have been identified. So that's why they kind of put that in there. And this substitution can do a lot of different things. It can make you make a different protein. It can be what's called a stop codon, so everything just stops after that. So depending on which mutation you have is probably why families tend to cluster in all breasts. You know, you'll see a BRCA family that's a lot of breast and hardly any ovarian, or a lot of ovarian, not a lot of breast. And everyone has it versus just a few of us have it, and why there's such a variability within BRCA of what you see in families. So this is just kind of my summary of why do we care that they have all these germline mutations or mutations at all. Well, because we can do something about it. We can do prophylactic surgeries. And there's also actual items for treatment if they do get cancer. This slide is actually from somatic data, so they're looking at the tumor and these same genes being mutated within the tumor, not necessarily germline. But you see in this study, about half of the ovarian cancer cases looked at had a mutation that we have some drug that has some action within that mutation to give them. So it can be very important. The biggest things we have right now for BRCA, we have PARP inhibitors that work better if you have a BRCA mutation for ovarian cancer. And then immunotherapy works better if you've got the microsatellite instability. So Lynch syndrome patients may have that. There's still a lot going on with does this affect their overall prognosis, and kind of yet to be determined. So in conclusion, patients should be screened per the guidelines for germline mutations in ovarian and endometrial cancers. Risk may be modifiable if found before disease. Specific mutation risks are constantly changing and evolving. And the results may affect treatments that we have now, and probably even more so in the future, as everything being studied is focusing more on specific mutations within the cancer. And here's my references.
I just have one. As far as the genetic testing and insurance companies, are you having a lot of issues with that, or it not being covered?
Yeah, so if they have a diagnosis of ovarian cancer, fallopian tube cancer, we really don't have any issues getting it covered. And if they screen positive with our MMR testing for risk of Lynch, we don't really have a problem. It's more the people who they don't have the disease. Maybe mom had the disease and she's passed, and now I want to get tested. So you're the next generation that you maybe have to do a little more work to get that prior authorized. But I'd say in general anymore, it's becoming easier and easier to get that approved. Even I've worked here for 6 years, and we had a ton of issues when I first started getting it approved, and now it's like nothing. You never hear about it anymore. So.
Your practice, when you're deciding between which biomarker to choose, between NSI and MMR, is there one that you prefer over the other, or do you want both?
So if we're screening someone with disease, our pathology department does the immunohistochemical for MMR. That's just how it's set up. If we're going to screen someone later in treatment that maybe doesn't probably have Lynch, but we want to know about that data to see if they'd be responsive to immunotherapy, it's a panel that's going to do both no matter what. So you don't have to decide anymore.